Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. We are grateful to you for being such fantastic listeners. Thank you for your continuous support. So far, we've launched 129 episodes since February 2020. Let's revisit a previous interview while we record new podcast episodes with your favorite GPCR scientists. Before we dive into this one, we would like to take a moment and thank our Dr. GPCR Ecosystem partners for their support, namely Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. Did you hear? We are working hard to bring you the Dr. GPCR University. It's a program where you'll be able to take a course on your favorite topic or even create a course to share with the community. For that matter, we are looking for experts to create courses on anything GPCR related and can't wait to unveil these to the Dr. GPCR community. Enter your course and you'll get a chance to win a cash prize for the most voted course in the ecosystem. Stay tuned and keep an eye out for our waitlist in the Dr. GPCR monthly newsletter. Become a Dr. GPCR Ecosystem Premium member and enjoy rewatching all the Dr. GPCR Summit and Symposia talks, the video cast of our podcast, and soon take a course at the Dr. GPCR University. You can also message members privately and network with our community from all over the world. And now, let's revisit this fantastic episode. Hello, everyone. This is Yamina, and I'm super excited today to have Kathleen Caron with me. Kathleen, welcome.
Thank you, Yamina. I'm so happy to be here.
I'm so excited to talk to you, although we finished recording 3 consecutive episodes honoring your dad, Dr. Mark Caron, with a large panel where you served as my co-host. So you kind of know where to go and where I'm going with the questions. So why don't we start at the beginning? And if you would like to introduce yourself, please.
Yeah, so I'm Kathleen Caron, and I'm professor and chair of the Cell Biology and Physiology Department, which is a large basic science department at UNC Chapel Hill, just a few miles down the road from Duke University, where my father was a James B. Duke professor of cell biology for well over 40 years. I've lost track of the number of years. And so yeah, it's been fun to, grow up scientifically in the same sort of research triangle park area. And my research program at first was not focused on GPCRs, and perhaps for good reasons. I tried to do my early training in different disciplines. I'm pretty far away, but as any listener of this podcast knows, GPCRs rule all, and so you cannot stray far away from GPCRs for too long. And so my research program has come full circle and is now fully entrenched in the world of GPCRs, and that was a really great thing for me, and a special relationship that I had with my father, given his impact on the field, and he's my father, he's my children's grandfather, and that was always the priority. But every once in a while, he was also an advisor and a mentor, and a great person to bounce off ideas and share great exciting stories about science.
I love it. And I always ask people, how did you get into studying GPCRs? But I'd ask you, when did you first hear about GPCRs? Because I think you may have a very interesting answer to that.
I think I do, yeah. So when did I first hear about GPCRs? Well, when they were first cloned. I was there. I was little, 4 or 5 years old, and remember those days very fondly. In Bob Lefkowitz's lab. Mostly I would sit in the administrative offices with the folks there, and sort of watch into the lab while all the biochemistry was going on. So I have memories, as a child of playing with lab reagents and some of the frogs that they would bring in, because they would isolate, the material from these huge frogs, and of course I would get to play with them before they would euthanize them. So I truly grew up in a lab, always in the lab, and what my parents would always joke that at a very, very young age, maybe 3 or 4 years old, I would most little girls cook with their mothers in the kitchen, right? They have the kitchen sink and they have the pots and pans and the bubbles and they're cooking. And my parents would say, Kathleen, what are you doing? And my reply at a very young age would be, I'm making cyclic AMP. And so my entire life I've been sort of focused on GPCRs and cyclic AMP. So, but I have to qualify that by saying that that's through the lens of a child, right? And
of course.
And so these were the things I grew up with, and I think most importantly for young scientists who consider a career in science, to me, I would hear those words and equate it with laughter and fun and excitement and passion. I didn't know what cyclic AMP was when I'm playing in the kitchen, helping my mom doing dishes, but I knew that it was important, and I knew my father loved it, and I knew that he loved his job and his career. And so, that passion and love for science, and just working hard and creating new things and discovering new things is something that both my parents instilled in the three of us and the three kids. So, maybe it's not surprising that I ended up as a scientist.
And it's interesting that you mentioned, ending up as a scientist. But first of all, let me take 3 steps back. I don't think there were many people in the world who were there as observers as these GPCRs were being cloned. So you have a special place in the GPCR world from that perspective. And I think it's an amazing one because you saw it with your child eyes, but then it got stored and it ended up propulsing you into being a scientist.
Yes.
The other thought I had is I don't know how many scientists in the GPCR field there are that have their children also working in science, not even less in the GPCR field. So I think you're in a very unique bubble.
Yes, it is. I think there are a few. I've met a few people, and I neglect if I cannot quite remember who they are. There are a few, father-daughter teams or father-son teams. No, but it is rather unique. And my father and I had several occasions where we were both at conferences together. Now as an adult, giving lectures. And one of my favorite ones, it might have been a GPCR retreat, the Great Lakes GPCR retreat, or maybe it was a Keystone meeting. I can't quite remember, but I think that the organizers wanted to have a bit of fun, and they put my father and I both in the same session. So although I am classically trained as a reproductive biologist and cardiovascular physiologist, I was speaking with neuropsychopharmacologists. But I think it was kind of a running joke, and I heard that there were bets in the audience of who would give the best talk, and so I think it was just fun for everyone to see the father and then the daughter speak. And
he went first, just to kind of add to your stress level there.
I cannot remember who went first. But I do know I gave a better talk. And I know that Dad was so proud of it. That was the fun part, right? That was so heartwarming. Throughout my career, he's just been my biggest champion, and he was happy and excited and wishful that I would give a better talk. And I'm teasing, of course. I can't give a talk as Mark Caron, but it was that love and support and just go get it, and go do it.
I love it. Well, I recorded, now that we're talking on this podcast and people will hear it later on, but I recorded a podcast with Bruno Giros. Yes. And Bruno had mentioned that your dad, Mark, would come to him and visit, and they would meet in France. And he would always talk about how proud he was of your research. And he was mentioning to Bruno, oh, I love the work she's doing. She's so great, and she's so much better. Like, her work is so much more interesting than what I've been doing. And I thought, there you go, Dad. That's a proud dad moment there.
That's a very proud dad, and a vastly overstated and exaggerated evaluation, I have to say.
But I think he was entitled to saying that, and Bruno was mentioning, and I was like, yeah, I remember. And while we were recording the first 3 episodes, 100 to 102, there was this fax, that was in your dad's wallet that was mentioned.
Yes.
And it was results from the work performed in Bruno's team. And I cannot remember right now what it was about, but your dad went to Paris and showed Bruno the facts, I think, still in his wallet, like 10 years later. And he said, this is the best results I've ever received. So I think there was that enthusiasm that came out towards his work, but also towards what you're doing.
Yeah, and I think Dad had that, that was one of the things in the episodes. Many people talked about how, the lab environment was always positive, even through the negatives, right? And that's something that's so hard to achieve as a PI or as a lab leader, or even just as a colleague and a member of a team. Because most of science is really hard and most of science is failure, right? I mean, let's just be real. 9 out of 10 experiments fail, 9 out of 10 grants are not funded. You have to have a lot of grit and a lot of patience, but through that you've got to find a way to stay positive. And that can come in many forms, and it comes with humor, and it comes with good relationships, and it comes with being able to detach sometimes, like step away and not let the bad experiment ruin your week, right? And I think Dad really, I think people really appreciated that humanism that he brought to the lab every day. And just we're all in this together. We're all going to work hard. We're going to have hard times, but we're going to laugh through it. And we're going to, because if you don't have that positive energy and that the humor to recognize, well, it's going to work next time, that it can certainly get really depressing, right?
Of course.
So I think Dad, certainly for me, he instilled in me that grit and that, don't give it up. And, it was hard and it's going to fail again, but keep doing it, right? And let's do it with a smile and let's have fun with it, right? So
you just gave me a great podcast idea to talk about the negatives and see who uses what kind of mechanism to kind of go over that hump of negative data or stuff that doesn't work. And I think that's an art in itself.
It really is, yeah. And as a department chair, I advise junior faculty now. And it's one of my favorite parts of my job, I have to say. I mean, I love science, but I also love mentoring young scientists and junior faculty, and we meet quarterly and we are talking about those difficult things, like when things aren't working, right? When things do work, it's wonderful, and everybody's, it's great and you celebrate, but it's those moments of difficulty and the struggles that you just have to have a lot of mentorship and a network of people to help support you and give you ideas, and approach that, as positively as possible, because that's science. If science were easy and we would always succeed, there wouldn't be science anymore, right? Because we would have figured it all out, right? So
you'd be measuring cyclic AMP in your kitchen.
That's right. That's right. It would be easy and we wouldn't have jobs. So the fact that it's hard is something that is not for everyone, but it's something that needs to be embraced. And to have a career as long as my father's, and still be, even, before he passed away, he was still, so actively engaged in grants and papers, and, the lab was really running at a very high level. To keep that motivation and joy takes, some really special talents and special insights about, what's important in life. And what's important in life are people's relationships, friendships, family, and humor and happiness and hobbies and, all of these things. So
agreed. Let's take a step back. You mentioned that while you were going through your training, you I can't remember the terms that you used, but you went into the science and worked on other topics before coming full circle back to GPCRs. And it sounded to me that it was kind of an intentional path, because you wanted to differentiate yourself.
Yes. I mean, absolutely, right? So when I trained in graduate school, these were still the days where there were not many women faculty, or at least there certainly were not many tenured women faculty. And so, and there were not that many women in graduate school yet, you know, so, and it was important to me that, you know, if I was going to be successful down this path, that I wasn't there because I'm Mark's daughter, you know, and I'm okay with it, you know, oh, it's Mark's daughter. I'm okay. I've been that way for 52 years. I'm Mark's daughter. But for my career, you know, it was important that my successes were not just due to, oh, that's Mark's daughter.
Yeah.
But that they were, you know, my own and of my own doing. That was even more important because I trained at Duke. And so, you know, even more important to really distinguish myself and go down a different path. And so when I was in graduate school choosing rotations, I was very deliberate to kind of steer away from, you know, GPCR labs, because that, you know, my father had such an influence. It was, you know, hard to find that niche that would be different, but that yet is something that I would still love and be passionate about. And so I met Keith Parker, who was my PhD advisor, and he was an MD PhD and chief of the endocrine division, and had worked on nuclear receptors, right? So, you know, growing up thinking of receptors, you know, this was something new to me, that there are nuclear receptors and they work on DNA, right? So no GPCR person's thinking about the nucleus and, you know, DNA. No. So this is perfect, right? So it's a different cellular compartment and there's these nuclear receptors, they bind to DNA, fantastic. There's not a GPCR in sight. And so, you know, yeah, exactly, right. So, and it was also developmental biology, which I was very interested in. And then lastly, Keith was just an amazing mentor. He was a father of 5 children, and was just, you know, had a great family unit, you know, of course, a wife who was so supportive, but also he was a dad and bringing his kids into the lab on the weekends. And so maybe in some ways that reminded me of my life, right? So, you know, I have really fond memories of having Keith's children in the lab on the weekends, because I grew up that way. But so he instilled in me this really the importance of work-life balance, and that, you know, family is important, work is important. And so he was just a fantastic mentor. And so, and in those days, we focused on nuclear receptors and a new protein that was identified in cholesterol transport within the mitochondria, and the process of steroid biosynthesis. And this was right around the time that knockout mice were really coming into being, right? So it was a big deal to make a knockout mouse when I was training as a graduate student. So that was my thesis, was to focus on these steroidogenic pathways using knockout mice. Yeah.
Yeah, I feel like there was this boom of, you know, we realized we can do knockouts, and then every conference you would go to, you would present not mouse data, and they'd be like, where's the mouse? Where's the knockout mouse? And you're like, no, it's different. We don't do that yet, or we're not going to do it, or we can collaborate. And then after you completed your PhD, what happened next? How did you pick, first of all, how did you pick your postdoc?
Yeah, so my postdoc, I interviewed at several places in the Triangle. That's because my husband is MD PhD, and he was still doing his training at Duke University. And so we're a dual career science couple, but we were never in that position where we could, in the early days, you know, pick up and move together, right? Because either I was in this phase of my training or he's in that phase of his training. So I did look locally, and again, very fortunate that the Research Triangle Park in North Carolina is just this wonderful, you know, ecosystem of, you know, institutions at NIEHS, NC State, UNC, and Duke. And so I interviewed at all of these different institutions, and really just fell in love with the research program of Oliver Smithies. So Oliver Smithies was here at UNC, and he is one of UNC's Nobel Prize winners for his contributions for gene targeting in mouse models. And so, you know, I knew of Oliver because when I was a graduate student making a knockout mouse, there were not core facilities that made these things. It didn't, like, it wasn't automated like it is now, and you couldn't go online and buy these things. You had to collaborate with people who were making mice, and Oliver, being, one of the first person with Mario Capecchi to characterize homologous recombination, he was the guy. I mean, he was the guy who made knockout mice. And so it was just this ability to take, this approach, a holistic approach of querying the function of a gene or a protein in a whole animal and modeling human disease was just like absolutely the most amazing, thing that I could think to do my career on. And so it was, wonderful when Oliver offered me a postdoc position. It was the lowest salary of all the other offers I had, but I took it because he had not won the Nobel Prize at the time. But I knew that he would be a great mentor. And I just loved this approach, right, of gene targeting. And so I learned myself how to do gene targeting, and that was my postdoc. And I in the end generated, I think, over 40 different gene targeted mouse lines, because we were a gene targeting factory, right? We made mice. Yeah. Now, of course, we ship it off. Even in my lab, we ship this stuff off to be done by companies. But that's where I learned really cardiovascular physiology. And that theme has continued into my research program. So we studied genes that regulate blood pressure and vascular tone. And we also studied genes that regulate heart development and heart injury. So I learned cardiovascular physiology with Oliver, and using this applied mouse models approach. So.
I love it. I love it. And then once you completed your postdoc, or did you already know that you wanted to be a professor? You kind of had a biased vision there at home growing up, but was that something that came to you now? Was it a natural progression, or do you think you were influenced?
Yeah, to stay in academia, was it a natural progression? Yeah. No, it wasn't. Like many people will tell you, there's bumps in the roads, and you consider different careers. Many of the postdocs from the Smithies lab, in that era, and I was one of the more senior postdocs before he stopped recruiting people. So Oliver passed away in late 80s, and so the lab was kind of getting smaller and smaller, and many of the postdocs who trained there went into positions in academia where they were running core facilities, right? I mean, if you're University of Wisconsin and you need to establish a new mouse core facility, you hire postdocs who trained in the lab that is credited for doing the technology. So a lot of the postdocs went into those types of roles, core directors, and many of them also went into industry. Doing the same thing. And I had several offers from industry. At that time, there were lots of companies that were popping up with the objective of, knocking out every gene in the mouse genome, or doing mutagenesis of the genome in mice. And so I was very skilled to maybe leap into industry with the toolkit that I had. But, academia was always very appealing to me. I really like the teaching mission, and I love grant writing. Isn't that crazy? I love writing grants. I just love it.
We should put together a grant writing workshop.
Yes.
I'd love that.
Yeah, I love writing grants. It's just my funnest thing to do. I mean, finding the time is a different thing, but I really enjoy that creative process. And, getting that logic. I'm actually a philosophy major from undergraduate. So that sort of logical thinking and, laying out, an experimental plan is something I really enjoy, that I may not have had as much in industry, as an academia. And so it was just very fortuitous that a new chair of the physiology department had just been hired at UNC. And his vision was to bring in more people who were studying the mouse with a physiological perspective, right? And using the mouse as a model for human disease. And so it's very serendipitous and very fortunate that I was a great fit for him and his new vision. And so I actually stayed at UNC. Again, another unusual thing, but total serendipity, right? I mean, like it's very unusual that happens. But
it is, but at the same time, I think it was convenient for your situation that applied to you. And I know we typically leave the inspiring young scientist segment to the end, but I think people need to think about what works for them.
Yes.
And this worked for you.
It did. And I did interview at other institutions, but UNC, I mean, it's a shameless pitch for UNC, but it's a wonderful place. I mean, I just cannot imagine, what an amazing institution to launch your career and to do science, so many core facilities and just a great network of cardiovascular researchers. So it was a great fit with a wonderful new chair, Jim Anderson, who I knew was going to ensure that I was successful, right? So I put a lot of, trust in him, and he sure, paid off. And he was a wonderful mentor to me and helped ensure that I could launch my lab and be successful. So.
Yeah. I really like it, but once we stop recording, we need to talk about this grant writing workshop. I've been thinking about putting it together for such a long time now, and I was trying to figure out, okay, who can I ask? But since you love writing grants, we will find the time to record maybe smaller increments. We can make a plan. But I think a lot of people would be very interested in that because I hate writing. That's the worst thing that can happen to me. I'm a lab person.
Yeah.
You tell me, Yamina, this is the question, or Yamina, come up with the question. I'm thinking about the experiments. I have tears in my eyes whenever I plot dose response curves, and they're beautiful. Ask me to write a grant is just it. I think the process can be very enjoyable, as you mentioned, when you have the time to build up your abilities to do it. You have to do it enough times, but unfortunately we get pulled into so many directions, and as you pointed out, sometimes it's difficult to find the time to do it.
Yes. I think for faculty, I know that faculty, myself included, and faculty in our department do complain a lot, finding that time and blocking off that time. That's time management, a whole different
which we can have a workshop around too.
Workshop on that too, exactly. But early on, you're right. It's grant writing. It's very formulaic, and it's there is a formula to it. There's a strategy to it, and it's all about providing really innovative, exciting ideas, but yet in a very structured document that the reviewers are going to enjoy reading, right? It has to be an enjoyable read, because the worst thing you can do is write a grant that they're reading at 11:30 at night and upset because they can't find the figure, or this is, doesn't make sense, or it's not well connected. So there's a lot of, it's called grantsmanship, right? Of just thinking about ways to structure exciting science within the confines of a, formulaic, grant proposal. And those two things seem a little different, right? Like being different and innovative and cool and cutting edge, but yet very structured. And so, there's strategies to do it, actually.
That's awesome. I can feel your excitement around this topic, so we need to talk about it. Let's go back to science. You've started your lab. You're working with mouse models. Where do GPCRs come back into the picture at this point?
Yeah, and this is so funny. It's serendipity, right? So during my postdoc, again, I mentioned that I was working on factors that regulate blood pressure. And my project basically is Oliver gave me his R01, and he said, here, you do this project, right? So it was a 5-year R01, and just spare me this little story, but you'll see very quickly where it goes. So the objective was to generate an animal model where we can genetically control, at the investigator's choosing, the level of renin hormone. renin is one of the major homeostatic hormones that regulates the renin-angiotensin system. And the reason that it was important to clamp renin was because any time that the Smithies lab or other labs all over the world would try to study hypertension in a mouse model, renin would kick in and reset homeostasis. So even though you know that, knocking out ACE or angiotensin receptor should result in hypertension, the animal is smarter than we are. And the renin gene comes in and resets homeostasis. So the objective was to generate animal lines where that homeostasis is under our control and we can clamp it and then introduce these different genetic mutations and ask what each gene's contribution is to the regulation of blood pressure. So the genetically clamped renin transgene, which is a single site insertion gene targeting approach, is what my project was. It was a tool, right? So there was no hypothesis there, right? So control renin levels. What do? You change the levels of blood pressure, right? It was not an inquiry into what does this protein do or how is this thing regulated. It was building a tool. And one day I was in the lab, I was about, 3 years in, it was going very well. I had all these ESLs and, it was great. And Oliver came into the lab, which was unusual because usually he would be at his bench. He always worked at the bench. And so you would go visit Oliver. But one day Oliver came and visited me. And he said, Caron, and he always called me Caron, Caron, you look bored. And I said, I was scared to death. I was almost vomiting. It was like, oh my gosh, I'm going to tell Oliver, yes, I'm bored because your R01 is boring. Right? So, right? Like how obnoxious, right? To tell a future Nobel laureate that his R01 is boring, right? But it was in a way, right? It was, tool development is important and it's important to create these innovative tools that enable, science and good rigorous science. But it takes away that excitement and energy and hypothesis, inquiry, right? To discover something new. So I told him, I said, I am bored, Oliver. And he said, well, tell you what, very well, British, very well. You're doing really well. So, why don't you come up with something and you can make a mouse, of your choosing. So you pick a gene and you make a knockout mouse, and you decide. My only caveat is that if you knock out a gene, you should at least try to make it so that the mouse is hypertensive, right? Because we're studying hypertension. So in a very unromantic and uninspired way, I went to PubMed and I typed in vasodilator, because if you want a mouse to have high blood pressure, maybe you can knock out a vasodilator and then it'll make it hyperconstricted. And there were only 3 papers published on the adrenomedullin gene that had just been cloned by a group in Japan. And so with only 3 papers, I said, okay, this is a good one. So I cloned the gene and made an adrenomedullin knockout mouse. Okay, fine. So, and that was very exciting and it had a great phenotype. And then while we were publishing this paper, there was a putative receptor for the adrenomedullin gene peptide. It's a peptide hormone. And lo and behold, it's a G protein coupled receptor.
Of course it is.
Of course it is, because GPCRs control it. And I remember calling Dad. I was like, oh no, you're not going to believe this. Like, guess what? The adrenomedullin receptor is probably a GPCR. And Dad, and he laughed, of course, right? Because everything's a GPCR, right?
Yeah.
So my lab then worked, or with Oliver, actually, very much with Oliver's help. He helped me clone CLR. It's a nasty gene with tons of exons and very GC rich. Unlike most GPCRs, it's a very complex gene structure. And so Oliver helped me make the targeting vector for CLR. And so we made the knockout for that GPCR. And then the ramps were discovered, right? And this is where serendipity, what can you do, right? I mean, it was this fascinating, science paper. Oh my gosh, there's these new proteins and they regulate different facets of GPCRs. Yeah. And you just have to go there, right? I mean, so the ramps were identified because they modulate CLR activity. And so that's where serendipity comes in, right? There was no way I could ignore the ramps. So my career then, as Oliver promised, right? Do something, create something and take it with you, and you can launch your lab with that. It was a great partnership, and I'm so grateful to him to have given me that freedom and given me that ability to, I started my lab with a whole cohort of ramp mice and CLR mice, and that's a credit to Oliver and his generosity. And many PIs won't do that, right? But he did. And he gave me that chance. He entrusted me to develop my own thing. And I wouldn't be here today if it weren't for Oliver.
I love it. It's a great story. So you were bored and now you work on CLR and the ramps.
And the ramps, yeah. And I remember Oliver, I was very hesitant about going into ramps first because I'm Mark's daughter, right? So,
yeah,
right?
Yeah.
That never goes away, right? I will always be Mark's daughter. So I was always very hesitant, like, oh, this is a big risk, to go GPCRs, right? And so that worried me. But the other thing that worried me is that the ramps were discovered through GlaxoSmithKline and Stephen Ford's group.
Yes.
And so I was just a little worried about competing or, having, a pharmaceutical company come in and scoop me basically, right? Because if you're going to commit to making knockout mice, you need to commit a year, of time before you're even thinking of publishing a paper. And there, Oliver also was just really inspirational to me. And he said, well, just give them a call.
And I was just going to say that. Why don't you collaborate?
And I said, what? And he said, well, just call this guy, what's his name? Stephen Ford, let just call him, ask him what he thinks. And I had to sit on that for a little while because as a young postdoc, you don't just pick up the phone and call somebody. So, but I did. And I'll never forget that day, right? I went to a little conference room off on the side where I could have some privacy. And I picked up the phone and I called Stephen Ford. And I said, hi, I'm just this little postdoc in North Carolina. And I'm wondering if, what you would think if I made ramp mice. And he said, absolutely, go for it. Do it. Absolutely. Let me send you my plasmids. And he did. He was so generous and was really kind and generous and supportive and said, go for it. We'd love for you to do that. And so, just branching out of your comfort zone and recognizing that the scientific community more often than not is collegial, not collaborative, not competitive, right? So
I love that story. Stephen was on one of our podcast episodes.
Yes.
And I remember, him telling the ramp story, and now I have another facet of it.
Yes. Yeah.
And he's such a wonderful guy. He's such an amazing guy. Loved it. I don't know if you had a chance to listen to that episode, but they had so for 2 years, this was during COVID. So for 2 years, he was mentioning that no one came into the house during the day. He's retired now.
Yes.
And on the day of, within the hour where we were recording the podcast, some roof workers were knocking. And he said, wait a minute, I have to go. So he gets up, lets the roofers come in and do whatever they need to do. And then we're recording, and then Fiona walks in coming home from something, and you can hear her heels and hi. And she goes in, and he's like, 2 years, no one came in at this time. And now that we're recording this is happening.
Yeah. So you got a 2 for 1. You got Fiona and Steve.
Yes, I did. I had Fiona previously, and she said, you have to talk to Steve. And
good.
I've had him, and they're both such wonderful people. I really are. Yeah, they're so amazing people, and I feel like it's important to talk about. You mentioned getting out of the comfort zone. It's important to get out of your comfort zone.
It is.
And you made an effort to get out of your GPCR comfort zone.
Yeah.
And then like the movie, the quote of the Godfather, just when I thought I was out, they pulled me right back in.
They pulled me right back in. Yes, exactly. There's no way to get around it. Yeah. but it's been wonderful, right? And I think that I never regret that I veered, into a different path. I thought that was really healthy and very good. but I love the GPCR field, and well, I just love the science, right? I mean, who doesn't? How can you not, right? But what a wonderful field, and just great people, extremely collaborative, and lots of great ideas, and so many things to do, right? So it's an honor for me actually to be a small member, making small contributions to this really big field. Yeah.
I think it's a great field, and you mentioned, people are being, most of the time people are very helpful.
Yeah.
And this is a message out to all the trainees. It's okay to pick up the phone, and it's okay to send that email, because most of the time you're gonna get a helping hand helping you out.
Yes, more often than not.
Than not. I think so too. So now I have to ask, because I ask everyone, what is
Okay.
your favorite GPCR? And it can be anything within the GPCR family signaling.
Right.
protein.
My favorite GPCR is the GPCR that people never thought worked with ramps, and what do you know, it does. And it turns out that that's a lot of GPCRs. any ramp interacting GPCR, particularly the unexpected ones, are just fascinating, right? Because to me, as we're learning more about the proteomic networks and the local interactors and regulators of the receptors, not just at the membrane, but in the endosomes, and as they're trafficking, these ramp molecules, these, Mother Nature's allosteric modulators are there, and they are doing things that we never considered. And it really makes you think, well, gosh, we have a lot of experiments that we should repeat, because maybe we should throw in a ramp. If this is a ramp interacting receptor, and we use cells that don't express ramps, we'll get a completely different, pharmacology from the receptor, or different signaling pathway, depending on the ramp that you throw in. So to me, any GPCR that interacts with ramps is very exciting.
I love it. That makes me think about, expression, like endogenous expression of the ramps. And you mentioned experiments. I know hex cells have some, if not all, the ramps.
It depends on the hex cell subculture. Yes. There are some hex that have no ramps. And then some that express more one or the other. Yeah.
And then you never know which one it is. And I think it's this variability and this horrible thing about hex cells, which, by the way, I love hex cells. I loved working with hex cells because they're easy to work with, and over CHO cells, for example. But then I had to look for the vial of hex cells that from which I could grow a culture that didn't have this or that expressed.
Yes.
Which I think it's very important to keep in mind when you're starting a new project. To verify that your gene of interest is not expressed in there.
Yes, exactly. Yeah.
Physiologically, are ramps ubiquitously expressed in tissues and cells, or?
They generally are, but not all cells have all ramps, and ramps are expressed at different levels in different cells, right? So you can have a cell that has only ramp 1 and ramp 3, but not ramp 2. Or some combination, right? There are only 3 mammalian ramps. And so then it's just a combination of relative levels of expression. And I think one of the more perplexing questions in our field right now, as we think about ramps, and well, there's 2 perplexing questions. The first is, what is the repertoire of receptors that they do interact with? And when we define interaction, are we defining a biochemical interaction by Brett that we see in a hex cell because we forced it upon the cell? Or are we talking about a native interaction, in a native level expressing receptor in a native cell? So which receptors interact with which ramps? That's a huge question. And our lab and other labs, of course, are tackling this. But then I think another, so maybe once you have that repertoire of GPCRs, and it's more than just the secretin family, there's many, almost every chemokine receptor interacts with ramps. once you have that information, then it's the more difficult question, which I, this keeps me up at night. And I'm not quite sure I know how to figure it out. But like in a given cell, let's say there are all three ramps. How and why does a receptor decide? It's not decide. Proteins don't make decisions,
Yes.
but how
give them personalities. So
yes, but how would it choose which ramp it's going to associate with, and or how does the receptor, is there preference or is there specificity for a receptor to preferentially bind to ramp 2, as an example for CLR in an endothelial cell, but bind to ramp 1 in a neuron? And because neurons express ramp 2 as well. So how does the CLR receptor know which ramp it should be binding with in the different cell types? And this is something that I don't think we have a good handle on. I know we don't have a good handle on. And, we're approaching it in different ways. Of course, we're approaching it with mouse models, tissue-specific deletion of the ramps, right? But again, that's very difficult to tease apart, because now you're asking a physiology question on a cellular pathway question. And then we're using, proximity labeling approaches and, trying to identify the interactome of the ramps. So anytime there's a new technology that's, enables us to ask that, huge question, our lab tries to jump on it as fast as we can. So, but I think that these are, without this information, we're gonna, trudge along a little bit, right? So I think we need to identify those key factors. First, we need to define the potential interacting receptors, and then go in and ask that very difficult question of which ramp and why and how. How is it one ramp over another ramp?
I think that's an important question, and I'll take it to the next level. Would a disease state change the interaction between a receptor and a given ramp, or would it abolish that, or how does that even happen? I think answering the first question is vital.
Yes, yeah, absolutely. And we think it does. We know that there are physiological conditions that we've characterized, mostly related to estrogen and its regulation of ramp 3, that do change the receptor that they're interacting with and the functions of those receptors. So we know that physiology, estrogen for one example, or cardiovascular disease like cardiac hypertrophy, can change the ramp interacting GPCRs and what those ramps are doing to those GPCRs. So, it's
how cool is this?
It's very cool.
I think it's really cool. It's just it's just such an well, we could talk about how cool the field is and how cool it's for hours for sure. If you had a magic wand and you could, make appear a new method that would allow you to answer the central question that you're working on, what would that method look like? It's a tough one.
It is a tough one. Yeah. It might be a little along the lines of cryo-EM, but even cooler, right? I would love to be the magic wand, really crazy science fiction, right? I would love to be able to see the receptor in the membrane of a cell, a real cell, not a hex cell, a cell. So see it in a native tissue, maybe labeled or, peppered and decorated with flags or things, in a living state with its interactome, right? So if we could visualize and see those interacting proteins, and again, I'm thinking of ramps and other related proteins, maybe more beyond the signaling, right? I mean, we're getting there with beta arrestin and G proteins, right? But more so that these scaffolding proteins and these other accessory proteins that kind of control where the receptor is and where it goes and whether it's engaged for signaling. And if we had a magic wand to just see that, that would be amazing. Don't quite know how to do it, but
I don't know either, but I think it sounds like a fun movie to be watching in IMAX, 3D, laser, you name it, and
Yes.
you're sitting there and
Yeah.
you can see the whole thing happening.
Yes, that would be very cool. Yeah. And a way to sort of like at the same time that you're seeing it, you're seeing new proteins coming in, like it's somehow decorating the proteomic network in that receptor at that time, and ramp would, how does the ramp being there change how that receptor is presented, would just be amazing. So, and of course, the cryo-EM structures, Patrick Sexton has made these beautiful publications with the cryo-EM structures. So we're there at a molecular level, but I would like to take it just a little bit and look at the cell in a living tissue or a living organism. That would just be fascinating.
Yeah. Well, structures are great, but it's just a snapshot. I think a collection of structures that allow you to follow these changes and conformation and have put this together would be really amazing. Then again, we're still talking about a magic wand scenario here.
Yeah.
Who knows? Who knows? Maybe in the future we'll be able to stabilize and take snapshots of a receptor interacting with various proteins.
I'm certain. I'm absolutely certain. Yeah. So, it's magic wand, but it's not that far off. I think we will see this. We will get there. I'm actually pretty confident of that. And then once we do, that will open the doors to new therapies and toolkits for ramp interacting GPCRs, so that and arenumab is the best example of this, right? Where the antibody itself, the first antibody to target a GPCR, by virtue of the fact that it is recognizes the epitope of CLR and RAMP1, right? So very, very specific to that receptor when it's with RAMP1, not when it's with RAMP2 or RAMP3. So, when we can have all that information, it will enable, drug companies to start developing highly specific antibody-based tools to target these receptors.
Yeah. I think it's cool. Do you have a favorite between ramp 1, 2, and 3? You mentioned ramp 2 multiple times. That's why I'm wondering.
Yeah. I really like ramp 2 because unlike ramp 1 and ramp 3, it's essential for survival. So if you knock out ramp 1 or ramp 3, the mice live to an old age and they're fine. They're not fine. They have phenotypes, but they live. Ramp 2 is critical. And as I mentioned, I've been trained as a developmental biologist, right? So I love development. So anytime you have a protein that is so critical that it's essential for life, that to me, that's like, that just, makes the hair on my back of my neck. Like, oh my God, it's so important, right? You can't live without it. So I really love the essential requirement of ramp 2 for survival, and how that, then in an adult animal plays out in multiple different organs with multiple different receptors. So it seems to carry a lot of importance to the function of the mammalian system. But ramp 3 is very perplexing too. I love ramp 3 because it seems to be doing things that we really don't fully give it credit for in terms of receptor recycling, constitutive activity of receptors that have perplexed the field for decades. Our data and some maybe yet unpublished, and some published, are really starting to shine a light on the importance of this ramp in these sort of unexpected roles. So I think ramp 3 is going to be fun.
I think so too. And well, I'll talk to you. I have a couple of ideas. I'll talk to you after we stop recording.
Awesome.
People are gonna be wondering, like, what are they talking about once they stop recording?
Yeah.
I ask this always, and so far the answer has been yes, but I'll ask it anyway. Do you think GPCRs are still good drug targets? And why?
Yes. Yes. I think they are, and maybe, alluding to what I just said, cheers to the small molecules and the drug screens, right? I mean, that's so key and so fast. And But, again, as we start to learn about how the receptor is complexed with other things at the membrane, right, or in the endosome, right, if we can exploit its neighbors to develop better or more specific drug targets that would be specific to a cell and not every cell, right? I do think that that's a new frontier. The cartoons always depict the GPCR in a plasma membrane, and there's nothing else around it. And that's just not the biology of it, right? So if we can learn to exploit its neighbors to develop these, tools, pharmacological tools, right, that take into consideration the receptor as it lives in a native cell with all of its neighbors. To me, that kind of blows open so many possibilities for drug targets, right? That it might not just be the receptor, but it might be it as it's complexed with other neighboring factors, right?
I think it makes a lot of sense. And I know we typically focus on the GPCR itself. And I wonder if we could think about focusing, comparing disease versus healthy state, and focusing not only on the GPCR that we know is implicated in that disease, but also on the changes in the neighboring proteins,
Yes.
intracellularly as well. And I like the idea of targeting the receptor on cells that have something changed.
Yeah.
So I think having this potentially, if we're thinking about antibodies, having antibodies that recognize, I don't know, RAMP and the receptor, and we know that RAMP2 associated with the risk receptor only happens in cases of cancer or, any sorts of diseases. I think this is a really, really cool. But then again, we have to learn more about who are the neighbors and what do they do in that specific disease state. Which I think opens up another can of worms, or we go down a rabbit hole, because then sometimes we don't have the tools, or we don't know what are these neighboring proteins that influence receptor function.
Yeah. And I think we have to appreciate that will differ from cell to cell to cell, right? So an epithelial cell will have a different, the receptor might be expressed and ubiquitously, but its partners in an epithelial cell will be very different than in a neuron or in a stromal cell, or, so I think the cell type is also going to change that as well, right?
There's plenty of space to write more grants
around
all of these questions. All right, let's talk a little bit about advice to junior scientists. We've talked about it a little bit during our conversation so far. Anything else you want to share with the audience that you feel like people should take into consideration whenever they're thinking about their careers? And I wanted to focus a little bit about life balance, life job balance.
Work-life balance. I think it's there's lots of advice for this, and but I think ultimately you have to have a good support system, and so you have to be aware and you have to foster those positive networks that will support you outside of science, right? So it might not be enough to just have, a great PI and a good lab. You might also need, a supportive spouse or partner. If you're at the stage of having children, you need kids who love that you work, and you will inspire them to love that, of course. But having a supportive family who is supportive of your ambitions and your passions, and that's not so hard to do, because when you're happy and supported, it shows, and those people who surround you and love you support you in that happiness, and that, so but it's important to nurture that and not take it for granted, because when things get hard, that's what's there to catch you, right? So science will get hard. You will stumble. You will have times in your career where things, it happens to everyone. No one has this like perfect career. No one. but the constant is the network of family and friends that support you to get you through that. And sometimes that's a lab family, but it's more often than not outside of science. And so I think that that's, in terms of work-life balance, that's an important element. And it can be a big group of people, or it could just be 2 or 3, right? Like every person is different, right? Your network and your support structure, it's not cookie cutter, right? And so, but just having that something to fall back on, so that when the days are long or things are not going great, you have that support structure.
I love it. I think that's very important. And I think it transpired, getting back to the podcast episodes we recorded with over 30 people. I think that's what came through. The messaging is that it is a big Caron family.
Yeah.
And there is that support structure, and there is that personal connection that was built throughout those years. And I have a feeling that you took that lesson from your dad and put it into your work with your lab, with the junior faculty that you advise, into making sure that there is that support system.
Yeah, it's a really important thing. And throughout all stages, right? Graduate students need a great support structure, and junior faculty need it as well. And even actually, this is an interesting thing that I've sort of found myself becoming pretty passionate about and learning about. I'm trying to learn a lot about this, but the decision of when to retire, right? So, if you're a faculty member and you go through your career stages, and for faculty and young faculty, they're just wanting that, achieve the tenure, right? It's the road to tenure and get your tenure, right? Just today, actually, it's a huge, we had 2 faculty in our department get tenure today. I'm so excited.
Oh, that's great.
Guess what? They still need mentoring, right? So like tomorrow, they're a tenured associate professor, but the mentoring doesn't stop. And the mid-career faculty, have science throws you all kinds of things, right? And it's those who have grit and who are adaptable and who can change and evolve. Those are the people who, stay in science and continue to love it, right? And so mid-career faculty often, have challenges of, well, there's always work-life balance, the little kids, it's little problems. The big kids can sometimes be big problems, right? So projectile vomiting at daycare is rather different than, teenagers who have a variety of different issues, let's say, or young adults. So the spectrum of family balance changes as our careers evolve. And then also scientifically, our career, as you progress in your career, technology advances much faster than you do. And so it's a challenge for mid-career faculty to stay abreast and stay on top of the new technologies. And sometimes it's difficult to recognize, what, I don't think I'm ever going to be able to do this. I don't know that I can code in R. Like this is past me, right? Like, or, I don't know that I can do this new fancy technology, but how do I, still embrace it and bring it into my laboratory? How do I stay cutting edge, So I think mid-career or established faculty have those difficulties, and they need mentoring, and it comes in different forms, peer mentoring or networks and things. And then there's the senior faculty who face a totally different question of when do I retire, and when is a good time, and how do I do that? Because when you're a scientist in academia, it is your identity, it is who you are, it is what you do 24/7 all the time, even on the weekends. It's like it's all the time. And then you come to a point where you're tired, or maybe you want to do something different, and how do you step away? So I've come to sort of be an advocate that mentoring, and support structures are needed throughout the scientific career. It's different flavors, it's different issues, but we should, mentoring is key at every stage of the career. So our department is trying to do that, provide different types of mentoring structures and resources for our faculty through the career stages, just to provide a supportive community for our faculty.
I think you made an important point here about mentoring, and I think it's all about, and I think even a 2-year-old needs mentoring. I think we need mentoring all through our life. And I think as we grow and we end up in science and we have our labs and things like that, we get to choice as to who do we want to be mentored by, or who do we want to mentor. But as a 2-year-old, you can't really pick mom or dad, or whoever is in your life. And it's important to also acknowledge your limitations and say, well, I don't know how to code in R. I know that this could be really useful. Why don't I collaborate, or why don't I hire someone who actually can do that?
Yes. Mm-hmm. Yeah.
And I think as academics, myself included, I grew up in an academic environment where I had to prove that I can learn and do this. And I think we're past beyond that. You don't have to be doing everything. You just have to figure out, what do you need? Is it really what you need? Can you learn it, or can you find someone who can help you do the work? And then give your head the space, at least in my case, give myself headspace to think about higher level questions instead of focus on the pipetting or on the coding part. Absolutely. Where I know it's never going to work. I would never, I can't. I mean, if my life depended on it, maybe.
Yeah. But I think that that's one of the nice parts that, most academic institutions now, I believe most, certainly here at UNC, have really embraced and recognized the importance of team science. Yeah. It is impossible to do science as an individual. Even as a lab, it's really challenging to be at the cutting edge of your discipline, and any discipline, without relying and receiving expertise that is interdisciplinary, right? So the best research, in my opinion, right now is happening at those borders where typically disparate fields are coming together and saying, look at that. Who knew that if you apply a mathematical model to, the structure of blood vessels in the skin, you could predict, permeability. Who knew, right? So, applying math and modeling, or just take these intersections of different disciplines that have traditionally not worked together, I think is where we're really finding a lot of really new and exciting insights into biology and medicine. And so team science is really, it's the answer to this, right? You can't be an expert at everything. You just can't. So having those great skills of being a good teammate, as my daughter is on a volleyball team and we talk about being a great teammate, this is a lesson, life lessons that are important to carry forward, and being a gracious teammate, and a generous teammate, and always giving your all to what you can contribute, in appreciation for what others share with you. So so team science is the way.
Exactly. I love it. And I think technologies, communication allows us to do that.
Yes. Yeah.
And I think that's very important. All right. I know we're almost at time. Last question. Okay. Top 3 aha moments that you had as a scientist that shaped your trajectory. It doesn't have to be aha moments as a scientist, just life aha moments.
Yeah. Top 3 moments that shaped my trajectory. Well, the discovery of the ramps. That's hard to avoid. So that was a big one. Let's see.
I think the cyclic cooking in the kitchen is a moment.
I think that's a big moment. Yeah. I think that I feel so fortunate that I grew up with that model, that science is so fun, right? How lucky am I, right? I am so lucky to have had my father and my family expose me at a young age, and because a lot of people don't have that. I meet so many people and they say, what do you do for a living? I'm a scientist. And they just look at me, I've never met a scientist before, so I think I am just one of the most fortunate people to have just fallen into, who I am, and I'm so grateful for it. So that's a huge factor in my trajectory. And then I think the other one that I have to say I think has been really something that I really value and enjoy tremendously is leadership and mentorship. And, when I charted my career and, and don't ever think that I don't want to give the impression that I had a plan or a path or that this would turn out perfectly, right? You just kind of stumble along and hopefully things work out, right? But somewhere along the way, it really became evident to me that I really enjoyed mentoring and I enjoyed academic leadership. And that's a tough decision, right? To say, okay, I'm going to take a certain portion of my time and dedicate it to the success of others in the role of a department chair, or and I really do enjoy that. And so it's something that I've so far have never regretted, right? That but it's a choice, right? To say that I have an administrative role, which I view more as a mentorship role. But learning about leadership, how to be an effective leader, how to be an effective mentor, is something that was a trajectory I had not predicted, right? If you had told me when I was a grad student or a postdoc that I would be a department chair one day, or a leader, and I was, oh gosh, no, never. What are you talking about? So it's an aha moment that sort of set a trajectory for me in academic leadership and mentorship that I'm very happy that it's happened, because I really enjoy that role.
Thank you, Kathleen. I was just thinking, like academic leadership and GPCRs, no, and you ended up pretty much there.
Yeah.
Thank you so much for your time. People can find you at UNC on the UNC's website.
Yep.
But people will also be able to find you in person at the next Great Lakes GPCR retreat held in November. More information coming out on that, and I am excited to meet you again in person at that meeting.
Yes,
I think the first time you and I met was also a GPCR retreat, and we were sitting at the dinner table, we were chatting, and I think it was that time right before the CXCR4 ramp, or the chemokine receptor ramp paper was being finalized, and I was excited because you told me about it, and I was like, I cannot wait for this paper to come out. Me being a chemokine receptor person. So I think it was a wonderful meeting you, and I want to thank you again for being my co-host.
You're welcome. Well, I want to thank you because, Yamina, it's such a wonderful tribute, and it's just going to have a wonderful legacy. And I just only wish my dad were here to see it. He would be so humbled and so appreciative. And so on behalf of my whole family, I really want to thank you for all your efforts that you've done. Yeah.
Thank you. It's been a great ride meeting all these people that your dad impacted, and we had to reduce the list.
We did. I know 30. There were too many, right?
I know, but I hope at least that everyone who's going to be listening to this, so the episodes come out by the time our episode comes out, 100, 102, 1 and 102 would have already been made public. But I'm hoping that people will, especially junior faculty and junior scientists, trainees, will get a feel and learn from the model that your dad put together. And I want to thank you again for all your help helping organize this, co-host these episodes, and I'll see you in person later this year.
Yes, that's right. Thanks, Yamina.
Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our guest and our Dr. GPCR team, namely Attila, Ines, Montserrat, Ivana, Andreina, Balint, and Julia. A huge thank you to our Dr. GPCR Ecosystem partners for their support: Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. You can connect with our partners directly in the ecosystem, so don't forget to join us today. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Soon, you'll be able to share the Dr. GPCR University. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.